Okay, so welcome to all the attendees for this Saturday webinar conducted by the IIT Alumni Center in Bengaluru. We are a group of people from all the IITs, who are very passionate about bringing the IITs, industry and governments closer. And nothing is more crucial right now than this issue of COVID-19. And part of our work has been in this lockdown situation is to bring experts to Janta via these webinars. And today is the fourth such webinar that we are holding, and we are excited about having two outstanding young people on the panel today. The first one, Dhananjaya Dendukuri, graduated from IIT Madras and then got a PhD at MIT. And he also received MIT's TR35 award and the Centuria Prize. So he's a very accomplished researcher. He came back to India and co-founded Akira Labs, which is a point of care medical diagnostics focused company. So, I think that's brought him very closely in touch with the COVID-19, and he'll talk a lot about that. We first met Dhananjay when he was at the IIT Industry Conclave 2020, and he was the rapporteur for our biotech session. And, he was so focused and so crisp. It was just a lovely representation of what happened during that session. Along with him, we have Professor Rohit Srivastav, Head of Biosciences and Bioengineering at IIT Bombay. Rohit also, I think, came back to India in middle 2005-ish after doing his PhD at Louisiana Tech University. And over the years, has become the head of bioengineering and biosciences. Again, needless to say, multiple honors, awards, and so on. And several patents as well. So we are fortunate to have both of you on this panel today. The format we suggest is that if Dhananjay begins and speaks for 30 to 40 minutes, and then keep the thing open for Q&A, and then Rohit and Sushila can, Rohit certainly can add a lot to what Dhananjay is saying, and between Rohit and Sushila, if we can moderate this, it will be great. Ideally, we would like to finish by about 6:30. For those who are listening and watching this, please put your questions in the Q&A bucket, not in the chat bucket, in the Q&A bucket. So that's constantly being monitored. And again, I'd like to say that we record all our webinars and they are put up on YouTube the following day. So those who missed it can also have an opportunity, and you can also share it with your friends and colleagues, the recordings. So without any further additions, Dhananjay, over to you.
All right. You can see my screen now, right?
Absolutely.
Okay, great. Thank you, first of all, Ashok, Kamath Mishra, and Sushila for inviting me to give this talk. I'm really honored to be here. It's interesting coming from the diagnostic world, talking about all of these things right now, because so many of these terms, like RT-PCR or immunoassays and immunoglobulins and so on, have suddenly become part of popular imagination in a way that none of us in these fields would have imagined before. And suddenly diagnostic testing has really come to the forefront because of the COVID-19 crisis. And so I really appreciate the opportunity to talk about this today. What I'm going to do today really is give you an overview of the kind of diagnostic tests that are being done for COVID. And for some of you who are very educated about the mechanics of how these tests work, this might seem a little simplistic and things that you already know. But I think for a lot of people who maybe don't know about the details of diagnostic testing, I've tried to make an effort to kind of explain a lot of the details of how these tests are actually made and what their sort of pros and cons are. And hopefully we'll elicit some discussion from all of you, because I know this is a topic that everyone is very interested in today, about testing for COVID and what the best tests are and so on and so forth. So I've structured my talk in the following way. I'm going to give you, since it's a science-heavy talk, there's a lot of jargon in biology, and it's important to understand that at least at some basic level. So I am going to explain a few terms before I actually begin with my talk, and then discuss really the two key types of diagnostic tests there are for COVID-19, which is the molecular, the gene tests, and the antibody antigen tests. And then talk about how these tests are developed and what sort of platforms are used in the labs to test for these things. So that's sort of the flow of the talk. We'll start, of course, with a picture of the virus that everybody has probably seen in some form or the other. The thing I'd like to state here is that the COVID-19 is actually the seventh of the coronaviruses that have been identified in human beings. So while there are lots of coronaviruses around in bats and pangolins and other animals, in human beings, the 2019 strain is actually the seventh strain of a coronavirus that has actually infected human beings. The most prominent previous one, of course, is the earlier SARS. But again, there's been MERS a few years ago. And there's even been some slightly less infectious common colds, for example, which also count as coronaviruses among this list. So as the name indicates, really, it comes from the sun, corona. And that is actually because of these spike-like structures that are very distinctive of these coronaviruses. And in this cartoon view, you can see these pink spikes. And this is actually a protein that is on the exterior surface of the virus. And that's really gives it this distinctive name, the coronavirus, right? So all of these tend to have this spike protein. In addition to that, there's a few other important structural aspects of these viruses. You have inside this spring-like structure that you see, and that's really the RNA, which is the genetic code of the virus. And that RNA is encapsulated inside something called the N protein. So that's what's shown here, the RNA encapsulated inside the N protein. And you have a few other structural proteins that are important for this virus to function, including the envelope protein, E, and there's also a membrane protein, M. So it's important, again, to understand exactly what RNA is, what protein is, in terms of actually designing some of these tests. So I'm going to spend a little bit of time on that. I think all of us know that there are the four building blocks of the nucleic, of DNA at least, are A, G, C, and T. And that's what's shown here. These four different chemical molecules are actually the building blocks, or the bricks, from which a more complex DNA strand is built. So here on this slide, I'm showing you DNA, which is deoxyribonucleic acid, and also RNA, which is ribonucleic acid. So the main difference between the two is that you have, instead of a thiamine, which is a building block inside the DNA, it's replaced by something called the uracil in the RNA. And that makes RNA actually a single strand, as shown here, as opposed to DNA, which is actually a double-stranded polynucleotide. So this virus here, the COVID-19, is actually an RNA virus, which means you have an RNA strand like this that is about 30,000 base pairs long. So if you could imagine a really long sequence of U, G, C, T's, 30,000 bases long, that's what's encapsulated inside this virus here. So the analogy, again, is that, you know, the bricks are the nucleotides, and then you could imagine sort of a wall being the next level of structure, the DNA, RNA, and then that all gets encapsulated into a much larger body, which is the virus here. Another term which is very important as far as diagnostic testing is concerned is something called the polymerase chain reaction, PCR, which won a Nobel Prize actually, and is almost 35 years old today. But the principle behind it is quite simple, although it's remarkable how it actually works so amazingly. And what it does is basically exponentially amplifies DNA using enzymes that are present naturally. So as we all know, DNA is really the key to life, and the body is able to continuously copy and replicate cells by copying, replicating DNA. So we kind of take over that machinery to do a polymerase chain reaction. So if you start here with a DNA molecule shown here as a double-stranded DNA, it's got something called a 5' end and a 3' end. But if you start with one copy of double-stranded DNA, and then you add what are called primers, shown here in pink, and just the building blocks, the bricks, the nucleotides, and put it together in a certain chemistry, you can actually go from just one copy to 2 to 4 to 8 and so on to 2 to the power of n as you keep increasing the cycle. So that's really the power of the polymerase chain reaction, is that you're able to exponentially amplify a small number of DNA or RNA to start with and produce enormous number of copies of it. And that will become relevant in a little bit of time when I talk about diagnostics. So the polymerase chain reaction, or the PCR, is the name that keeps coming up. And the thing to know about it really is that, like I said, it can exponentially amplify these nucleotides. The other sort of jargon I'm going to throw at you to begin with is the other important macromolecule, a long molecule, is proteins, of course, inside our system. So the spike protein that I showed on the virus is a protein, of course. And proteins are composed of amino acids, and there are 20 amino acids. So you have 4 nucleotides that make up DNA or RNA, but you have 20 building blocks to make amino acids. And it's not important to know what every single one of those are, but just important to know that each of those actually come with a symbol code as shown here. So A, G, I, L, etc. So you can actually, basically mention a strand through its entire sequence of these amino acids. And amino acids have a more complicated superstructure. So if you have a string of amino acids like this, they actually then will fold into all sorts of complicated ribbon-like or sheet-like structures, which then go on to becoming something like a spike protein. So again, using the same analogy, you have the building blocks, the amino acids, going all the way to quite a complex macromolecular structure, the spike protein. Again, this structure and exactly how it binds to cells in our body and so on is obviously very important to the way the virus infects and even to the production of antibodies, as you'll see in a bit. So this is a little bit of terminology before I actually get to the diagnostics part of my talk. So what is this coronavirus and how is it related to all of the other coronaviruses around, right? So this is a family tree, much like we all would know. We've all seen our own family trees, of course. But you can see here the sequences shown in red are the 2019 coronavirus. And the sequences shown on top here are the SARS coronavirus, right? So it's actually only about 80% as similar. If you compared all of the nucleotide strands from end to end, it's about 80% similar to the SARS coronavirus. It's much closer to some bat coronaviruses that are not yet seen in human beings, but have been isolated from bats. And then there's also the MERS coronavirus and some of the other coronaviruses that you can see in this sort of phylogenetic tree or family tree, effectively, of these viruses. So they're all related in some ways to each other. For example, many of them have the same spike protein, the N protein, the E protein, but the sequence of those could actually change from virus to virus. So what happens when someone's unfortunate enough to get infected by the COVID-19? So this is just a picture of your lungs. And really, as you get, the lung has this fractal type structure reaching to the very end. You have these sacs, these alveoli, which are where gas exchange actually happens between your blood and the lungs. So you breathe in air, you want oxygen to actually go into your bloodstream, and you want to take away carbon dioxide. So that's what's shown here. And these alveoli actually contain some important cells, type 1 and type 2 alveolar cells, which the virus actually binds to an ACE2 on the type 2 alveolar cells. So that's how it kind of attacks the lungs. So when the virus gets in through an airway and actually gets all the way in here, it can bind to the ACE2 on type 2 alveolar cell. And actually that leads to the building up of fluid, problems with breathing, and things could get much worse for people that have severe problems here. But the point that I'd like to mention here really is that the virus is present inside your lungs. And this is also something that I guess that we all know. So what happens when you actually go through a COVID-19 infection, right? So shown here is actually a progression of 3 important molecules. One is just the viral RNA, or the virus itself, shown here in blue. So you can imagine it keeps increasing over time, and then as the patient begins to recover, it kind of falls off. And of course, these numbers are average or typical numbers. There are better and worse cases, but just think of these as averages. Within about 7 days of the virus actually entering somebody's system, you could actually start the antibody production, which is part of your immune response. And this actually first comes up as an IgM. It's an immunoglobulin M. So this is an antibody that is released, that is made by your own body to combat the virus. That's shown here in green. So it peaks and then again falls off. And then you've shown here in red is the immunoglobulin G, which is what is typically confers long-term immunity. And we'll see that there are some complications associated with COVID-19, but typically this IgG actually confers long-term immunity and builds up, say, 14 days and onwards, and actually maintains quite a steady presence in your blood, sometimes for even years after you've passed an infection. So this is sort of the progression of different parts of the virus and antibodies inside your system. So you could imagine now, if you are someone that just knew all this information right now, and you said, okay, I need to develop a test for COVID. There's 2 obvious things to do. You could either look for a protein, and the virus has a few different proteins, as I showed you earlier, or you could look for the RNA of the virus. The RNA of the virus, or the molecular test. So it's unfortunate this name molecular, because everything is, I guess, a molecule. But molecular tests have a specific meaning in diagnostic testing. They refer to nucleotide tests. So gene tests or DNA, RNA tests are called molecular tests. These tests have become the gold standard for many infectious diseases. And why is that? Because in the early days of an infection, so when you go back to this graph a little bit, in this 0 to 5 or 0 to 7 days, you may have very, very small numbers of copies of the virus inside a person. Let's say maybe just a few tens or hundreds of them even. And the only way to actually pick that virus up is by exponentially amplifying it, like I showed you earlier, the RT-PCR. So you can't pick up something as small as 10 or 100 copies by itself. The only way you can do it is by amplifying it, say, 1 billion-fold. And that's exactly what PCR does. So let's say you did 35 cycles of PCR. That's roughly 2 to the 35 times, and that's somewhere in the billions. So you could go suddenly from 10 or 100 copies to billions of molecules of RNA. And that's what enables you to actually detect it using an RT-PCR. You could, in theory, also detect a protein directly. So let's say you have the S protein or the E protein of the virus. You could detect it directly, but the problem here is that you can't amplify a protein like you could amplify RNA or DNA. That's just chemistry. That's the limitations of chemistry. And because of that reason, it is preferred, especially in the early stage of an infection, to do a molecular test, and it's a lot more sensitive. However, as the disease progresses, your body starts producing this amplified antibody response. And for each copy of the virus, you could be producing thousands of copies of this antibody. So that's actually can be picked up quite easily, which is why 7 days or so and after, you can pick up a lot of antibodies with the traditional detection methodologies. The final point is, when you get to a post-cure stage, you actually pass the virus from your system. So the virus isn't there anymore, but the antibody signature stays on. And that's why you could potentially pick up people that have actually had the virus, responded, and now have this antibody response inside them. So you could actually find these people that the virus has passed through using the antibody test. So you could see that both these molecular, these gene tests, as well as the antibody tests have their own uses. Some more terminology that's important to define in diagnostics. This is sensitivity and specificity. And this is very, very important because these terms are sometimes used very loosely. So I've given this in terms of a very small example. Let's say you had 1,000 people in a population that you were studying, and 100 of them actually had COVID, all right? So you have 1,000 people to begin with, 100 COVID positive patients, and so 900 of them don't have COVID. You have a test which is picking up 90 out of those 100 people and classifying them as being positive with COVID. but it also falsely classifies 10 of those 100 as negatives. So that's 10 false negatives. Of the 900 people that don't have COVID, you classify 850 of them correctly as being negative, but you classify also 50 of them as being falsely positive, right? So no diagnostic test is perfect, as you can imagine. And you always have these kinds of tables that you would do from a population study. So the definitions of sensitivity and specificity are right here. It's quite simple to understand. So 90 divided by 90 plus 10, so that's 90% sensitivity. And specificity really is how many people do you correctly classify as being negatives. So that's 850 divided by 900. So that's about 94%. So an ideal test would have, say, 95% sensitivity and specificity. But right now, in fact, many of the tests for COVID probably don't have this kind of performance. But since we're in a hurry to get something out, there's a lot of tests out there that probably don't have this kind of performance. But if you give tests enough time and optimize them, you will get to this kind of performance eventually. Another term that's very important, in addition to sensitivity, specificity, something called positive predictive value. As you can see from these numbers, there's a fairly large number of people that are false positive at a population level, right? So there's 50 out of 1,000 people actually think they have COVID, even though they don't, although the test is 94% specific. So the other important thing is actually something called positive predictive value, which is if you were classified as positive, what is the chance that you actually have the disease, correct? And that's different from 90%. So that's actually 90 divided by 140, right? So that's closer to maybe 60 or 70%. So that's very important, especially at low prevalence. So if you only have 1% of your population that actually has the disease, you could end up with positive predictive values that are much lower than 90%. So this is very important for planners and epidemiologists and so on to understand the value of a positive test. So let's go to a little bit more of technical detail now about how we would actually design one of these molecular tests. So let's assume that you want this very sensitive early detection to happen, correct? So you want to pick a gene or a set of genes that you would like to test. So here I have a very condensed view of the genome of the COVID virus, right? So you have about 30,000 base pairs, and they're arranged here from 5' to 3' end. I've just shown some random sequence. By the way, this is just me typing in those things there, so this may not be exactly correct, but just to show you that that's how it would be arranged inside there. And what people have found, because now coronaviruses have been studied for about 15 years, so people know quite a lot about them. And like I said, they share many of these features. So they share the spike protein, they share the membrane protein, they share the E protein, and so on. So you might decide that you would actually target, let's say, the spike protein, and you would want to detect the gene for the spike protein as your PCR test. Or you might want to do the spike protein, the E protein, and the M protein, let's say. So some of the good PCR tests actually are trying to detect all three of them and not just one of them. But if you target any one of them, the next thing that you would do is actually design a molecular assay to do that. And like I said earlier, the way to do this is using a very standard PCR kind of chemistry. There's a variant of it called TACMAN chemistry that's used for these kinds of assays, where you have a primer, as I showed here. So this is your original strand, all right? And a small detail that I missed, I guess, is that you have to convert the cDNA. So you have to convert the RNA into DNA. So once you do that, you have this strand of DNA, and then you have these priming sequences shown here. You have a forward and a reverse primer that actually bind to the DNA. And you have also these TACMAN probes that bind somewhere in the middle of it. And we'll come to that in a second. These are some of the sequences of these primers that were released by the US CDC, China, and Germany. So this is just to give you an idea. And this is something, honestly, that anybody with a computer sitting at home could actually generate their own primers. It's not all that difficult with the sort of bioinformatic tools that are available to us today. It's about finding a sequence that binds very specifically to a part of your gene, and then letting the DNA polymerase do the rest of the job and build that gene all along, basically. So these were the three primers that were released by the US CDC, and they took back this one actually, N3, as being not a very good one and causing sort of some false positive results. So they're sticking with N1 and N2 in the US. You can see that people have used different genes. So the US CDC is recommending the N gene, Germany is recommending the E gene, N gene, and the RDRP gene. So there's different ways to do the same thing. And it all comes back to which of the genes on the virus do you want to amplify? Is it 1, or is it 2, or is it 3, or is it, you know, all of them together? So that's what's done. Now, RT-PCR actually stands for reverse transcription polymerase chain reaction. As I mentioned to you earlier, you actually start with an RNA and not a DNA. So you need the small step of actually making the RNA into a DNA, and that's called reverse transcription. So what you do is you first collect the sample from the patient, then you extract the virus from that sample, then you convert this viral RNA to DNA, then you amplify the DNA, like I said earlier, and that's converted into a fluorescence signal. I'll tell you how in a little bit. And then you analyze those results to tell how much virus is actually inside a patient's sample. So let's look at each of these steps a little more closely. And this is something that's very, very important and not talked about so much, is how you collect samples from patients. So here I have four different spots from which you could collect samples for COVID-19. The first one here is what's called the bronchoalveolar lavage. It sounds a little painful, but basically you have to stick a tube inside somebody's lung and pull out that fluid that's accumulated in their lung. Especially for patients in the ICU and so on, you can imagine that this might be the only way to actually get sample from them. So this is bronchoalveolar lavage. The other is sputum. Sputum is basically getting people to cough out from their lungs this fluid effectively, and it's used often in TB. So that's sputum. Then you have a nasopharyngeal swab. So basically you insert a swab into someone's nose and you collect the snot from there. And then you have an oral swab, which is sort of the easiest thing to do. You just stick a swab into somebody's mouth, and the back of the mouth might contain some of the virus, and you pick it up, right? So one important point to note here is even these swabs are actually special purpose designed. They're not like your Q-tips that you might use, for your ears, for example. These swabs tend to be very bristly, and they actually have the ability to, what you want to do is be able to pick up as much virus as you can. So even something like the swab has to be optimized, and you can't just use any material to do this. It's very important what the swab is and how it's done. And as a small aside here, there's only a couple of companies in the world that actually make these swabs really well. And as you can imagine, they're completely run out of them. So one important point I'd like to make in this table is this is actually from an RT-PCR test that's being performed right now in the clinic. That depending on the sample that you take, the sensitivity of the test actually changes. This is very, very, very important, because what it says is that you might have the best test in the world that's amplifying the gene, that's doing very sensitive detection and so on. But if you messed up in terms of how you collected the sample, then no good test can save you. This is what's called a pre-analytical error. So as you can see, the bronchoalveolar lavage, not surprisingly, because you're getting all the way into the lungs, has the highest sensitivity. And the oral swab actually has quite poor sensitivity of only about 32%. So it's very important in terms of how you take swabs from the patient. And like I said, this is something that is being done maybe at people's homes or at camps or wherever. So training people to do this properly is also very, very important. The next step after you collect that swab is you put it into some medium which actually inactivates the virus immediately. That's obviously very important because it's a very infectious sample. And then from that medium, you need to extract the RNA. So what that means is you have to break open all of the cells there, including the human cells, the viral cells, etc. That's called lysis in biology. So you break open all of that and then release the RNA. And you release not just the RNA, but you release DNA and all sorts of you might have, for example, a bunch of human cells there which will release their own DNA. But there are these columns which actually will bind very specifically to the RNA from the virus, as shown here. And then you actually wash off all of the other stuff, which might be various contaminants and cellular debris and so on. And you wash it off, and then you elute or remove the DNA, or the RNA rather, from that column. And that's your starting material for the PCR. So these are very, very important steps. And these are done in a large lab in a very automated way. So they would have a liquid handling robot that is basically doing this sequence of steps and actually producing this eluted RNA. All right. The next thing that you do is actually, like I said earlier, run these TACMan assays. And it's quite simple. As I said earlier, you have to reverse transcribe the viral RNA into DNA and then run this amplification reaction. The important thing to note here is that in addition to these primers shown here, you have also these TACMan probes. The TACMan probe is a very special molecule. It has a fluorescent marker at one end and something called a quencher at the other end. So when it's in its normal state, it is not fluorescent because the quencher actually prevents this molecule from fluorescing. But when you run this TACMan, this polymerization reaction, what happens is that the DNA polymerase, which is the molecule that actually makes your DNA, and there's a special something called a TAC polymerase that's used over here. It's a heat-sensitive polymerase that comes from a special, from something called Thermus aquaticus that was found in a hot spring many years ago. And it's a very important molecule because it can function at high temperatures, unlike, let's say, polymerase inside our own body. But what it does is, as the polymerase forms this complementary strand of DNA, it cleaves off this fluorescent molecule, creating fluorescence. And as you amplify right now, you go from 1 to 2 to 4 to 8 shown here, you keep increasing this amount of fluorescence. So that's very, very important. And this amplified product, this fluorescence can be detected by an instrument. So there's a thermocycler and there's a fluorimeter. A combination of these two can actually pick up the fluorescence. And the fluorescence has this very standard S-shaped kind of signal where it's kind of, there's a lag phase, there's an exponential growth phase, and then it eventually plateaus. So all of this is done automated. So what you do actually is run a number of cycles where you actually do a combination of denaturing and annealing the DNA. What that means is that the DNA actually in its double-stranded form at say a lower temperature, about 50, 60 degrees Celsius, is heated up to 95 when the double-stranded form separates into the single-stranded form. And that allows you to then actually create more and more templates, which you can then anneal and copy as you go along. So this is actually a standard qPCR curve. And what you do when you're setting up any of these instruments is you actually make sort of a calibration curve where you'll have put in different number of virus copies, right? So you will artificially spike in, let's say, 10, 100,000, 10,000 virus copies. And each of those will have a different plot that looks something like this. So as you can imagine, the one with the lowest concentration of virus will actually take the longest number of cycles to amplify, whereas the one with the highest number of virus particles to start with will amplify quite fast. And in between, you have all the intermediate concentrations. So you can use that to make a calibration curve like this. So when you run an unknown sample, that will give you, let's say, some shape like right here, and that will allow you to say how many copies of the virus were in that unknown sample. So this is a quantitative PCR reaction, and this is how you use a calibration curve to then back calculate how many virus copies there are inside a known patient sample. So this is a very typical test interpretation. Let's say that you have a test that is looking for N gene, E gene, and the RDRP gene. In fact, this is a very standard one. I think this is the one that's being used right now in India. This is RNA-dependent RNA polymerase. It's another viral gene that's present. So if you had all three as positive, you would then classify that patient as COVID positive. And you'd also always run a human control to know that you're extracting your DNA and RNA correctly. But the more complicated cases are when, let's say, you have only two out of the three of them as positive, or only 1 out of 3 of them as positive. You might come up with some chart like this, where when you say only 1 out of the 3 genes is positive, you might say ambiguous or test again. And of course, if all 3 genes are negative and the human gene control is positive, it's a negative result because none of the viral genes are amplified, only the human genes, which you expect to be in a sample that's coming out of the human, are positive. And if all are negative, then it's a problem with your test protocol. You need to retest. So this is your very typical molecular tests and how they're interpreted. Maybe this might be a good point to actually pause because the next part of the talk is about the immunoassays and the antibody tests.
Yeah, I was going to suggest that, Dhananjay, because I'm seeing some questions in the QA section, and I was thinking maybe we should just pause for a minute here, maybe 5-minute break, in which let me ask you some of these which have been posed in the QA section.
Sure.
Okay, so the first one was was the 1917-1919 Spanish flu a coronavirus?
I don't believe so.
Yeah, I don't think so either.
Yeah,
I think it was a different class, influenza.
Yeah, it was an influenza virus. That's
as
right.
I remember.
Yeah.
So the other one is when you were discussing this the slide 12 on sensitivity, specificity, positivity, negativity, that stuff.
Yes.
There was a question: is it the same as concordance amongst positive and negative? I don't think the question was complete, but I guess there was another question in the chat box. Let me just see that question. So the question was: isn't sensitivity 90 over 140 and specificity 850 over 860?
No. So okay. So the way that the sensitivity is calculated, maybe I'll just go over the logic again. Sensitivity basically is all of the true positives as measured by some gold standard, right? Let's say the gold standard in this case is a clinician has looked at their symptoms and their chest CT and so on and said this person is 100% COVID positive, right? And if you know that information and those 100 patients you run your test on, and how many of them is it picking up? That's sensitivity basically.
Right.
So that's.
And how about specificity?
Yeah, specificity.
So there is some confusion in the specificity.
Yeah, I guess specificity is sort of the inverse, where the people that don't have COVID, same way you have 900 people in the population that don't have COVID, measured again by some gold standard. So the key point here is these are true. 900 is sort of the true number of negatives, right? Known by some gold standard, and you are only classifying 850 of those 900 accurately, and you are missing. You're classifying 50 of them wrongly as being positive, although they are negative. So that's where your specificity comes from. 850 divided by 900.
Okay. So I guess that's there. So there was another question. I have a few which I have been noting down, but let me just run through the QA first.
Sure.
So in the QA, there's another guy who asks, do we have capacity to make swabs in India?
Yeah. So this is the thing. I think it goes back to this thing about what is a swab, right? So like a Q-tip kind of a swab, if you looked at it under a microscope, that is like what you use as a earbud, it looks like a non-woven matrix, okay? But if you looked at a Copan swab, which is being used for the RNA extraction, it will look like bristles, right? All of them will be standing like, how when your hair stands up when you're like alert, it looks like that. That's what RNA extraction or DNA extraction swab for these kind of samples would look like. So they're made by some sort of electrostatic precipitation. And so Copan in Italy, as it happens, is actually one of the world's most famous companies that make swabs for all sorts of tough samples, including stool and things like that. So you need to have this electrostatic precipitation figured out. That's what I know about these swabs. And I don't know if anyone specifically is making things using that technique in India.
But I guess, now is the best time to bring most of these technologies, that we have been dependent on, China to bring to India,
yes.
Right?
Absolutely, Rohit. I couldn't agree more. I think this is the time that we all know we have the capabilities. And if there is, I guess, a long-term policy that we are looking at sourcing a lot of these things locally, which we should, I think it's absolutely the time to do that.
Yeah. So there's another question, so when you went to this RT-PCR slide, I guess this was slide 20, 21, somewhere around that, I guess. And you started talking about the reverse transcription process. There is a question which says, how does RT in PCR distinguishes virus RNA from other RNA? And I guess you answered that, right? Because you said that you do separate out the other DNA samples when you were taking the samples, right?
Yeah. So, but maybe Rohit, the question is, let's say that the person is infected with 3 different strains of a virus, right? So
Right,
maybe
right,
they have, COVID-19, and they happen to have some other virus like the common cold, correct?
right, right,
The answer is that you would extract also that RNA. Okay, so you would have both COVID-19 RNA and the common cold RNA, but your primers will be specific only to pick up your COVID-19 genes and not the common cold genes. So that's where the specificity.
right, right. Okay. So there's another question. What type of fluorescent molecules are used in the TACMan probe?
Yeah, so there is an entire family of these. That again, these are all sort of very, closely held, I think, chemistries made only by a few companies in the US and China. So they have these names, right? They have like hex, tet, fam.
Right,
So at different wavelengths spanning the entire sort of UV vis from
right,
400 to 700, you have different combinations of TACMan dyes and quencher probes. And again, a great knowledge to build in India if we can build that out in companies, because certainly it's a big problem now to get these kinds of things.
right. So another question is, I think that was well answered. What is reverse transcription? What is reverse in it? So I guess that's going back to the basics.
Okay. By the way, Rohit, somebody has answered here that that's a company in Chennai called Kriya Medical is actually started to manufacture these swabs.
Oh,
Vasu
that's
has
very
been,
nice.
which is
Very
great to know.
nice.
Thanks for the information, Vasu.
We are seeing a lot of questions. Yeah, that's true. So this reverse transcription, yeah. So what is reverse in it?
What is reverse in it is, I guess, well, you are going from RNA to DNA, correct?
Yeah.
And so there are enzymes which actually do that. In fact, there's an enzyme called reverse transcriptase that I'm showing you in this slide here. So you have this reverse transcriptase actually makes DNA from RNA. And the reason that's important to do is that all of these chemistries work very nicely for DNA. So this sort of annealing, denaturing chemistries work very nicely for DNA. And hence it's important to go from RNA to DNA rather than like doing it directly.
Right. So, I think there is a lot of questions around this fact that, if the virus mutates, what will happen, to the RT-PCR tests? Are they going to, you'll have to change something in that?
That's a very good question. That is a very good question. And I think, the short answer to that question is if it doesn't mutate too much, you probably will be fine. But then if there are significant changes and you happen to be picking up that gene, let's say the N protein gene mutates. In fact, by the way, it's much more likely that the spike protein mutates. It's well known right now that the spike protein is very, very variable, right? And that's sort of the smart part of the virus in some sense. That's continuously figuring out and optimizing itself. Which is probably why many of the PCR tests are going for the more conserved, genes like the N and the E gene. But even those could mutate. I think these viruses mutate every cycle, right?
Yes.
And so it's important to keep track of it. I think there are a few genomes now published from India, by the way. I was hoping to share some of that, but it was not enough time. So I think it'll be important to keep track of that through sequencing studies. And if there are significant changes coming, and those will get published and uploaded into this NCBI database, then you will have to change your primers effectively to kind of catch those changes.
And how easy is that to change primers?
I think that's quite easy. It's probably the more difficult part is, I guess, knowing when the virus has mutated and, how many strains there are. And that kind of thing is probably more difficult to actually know. But in principle, I think doing the primers for someone who's experienced at this is quite straightforward, I believe.
Yeah, and I was also hearing that, the virus has already started mutating. So we are seeing different, viruses now, sequences of the virus now. So, I think there was a study from Pune, which has identified 4 or 5 of these, of which one is the prominent one, which is showing up in all samples, but there are other ones as well.
I see.
Then there are other questions, but some of them will come to when you start discussing the antibodies and their testing.
Yes.
And so a question on the RT-PCR: does low sensitivity by source of sample, which is naso or oral, mean more PCR cycles can compensate?
That is a very good question. The answer is that not beyond a point. So for instance, the number of cycles that is commonly accepted as a sort of upper limit is about 37 or 38. If you do more cycles, you risk sort of amplifying other things, which are, contaminants and so on. And so that's kind of quite a strict number of cycles there. So people would not, if they don't see the virus in say 37 cycles, you would not go beyond that.
Right. And just a question from my side. I know there are several others, but we can probably take them later. But I had a question in the sense that you talked about this RT-PCR, but there is also something called RT-PCR, right? The room temperature one. So what is that? Or the LAMP assay? Which
Good
is
question.
being now proposed as an alternative.
Absolutely. Very important question. I purposely, I guess, did not talk about that so that I didn't confuse people too much. So RT-PCR, which I talked about, is sort of, you could call it the reference standard, the most mature technology. And in fact, nearly all the labs that we all know are running this technology. So I chose to talk about it. But what Rohit just spoke about, the LAMP method, it's called the loop-mediated isothermal amplification. That is a very interesting technology that does not involve thermocycling. And thermocycling means you need more complicated instrumentation to keep maintaining these two different temperatures. Whereas with LAMP, you could amplify by just keeping at say 60 degrees Celsius. So people are actively looking at LAMP to make these tests also. And to my knowledge, I don't know how many commercial LAMP tests are already out, but there certainly are going to be some coming in the next few months because it could be made a lot simpler with LAMP. The LAMP primer design is a little more complicated.
So, we are also seeing many of these. The reason I asked this question, Dhananjaya, was that you are into the same, diagnostics, and we are seeing several companies now propose a LAMP assay, and a LAMP device, an affordable LAMP device, because an RT-PCR costs about 3.5 lakh rupees. So it may not be feasible to put an RT-PCR in all of these, primary healthcare centers. So, that could be a way forward.
Absolutely, Rohit. In fact, RT-PCR, by the way, is nothing less than 10 to 12 lakhs. I know.
I know. I was thinking of the really cheap version that you could get. But anyway, So
you're absolutely right. I think if you could get a a LAMP reaction going, nothing like it. Ideally, a multiplexed LAMP that's able to pick up 2 or 3 of these genes, I think would be great in the Indian context as a sort of fast and quick and dirty sort of amplification and nucleic acid test.
Right. Okay. So, there are some other questions. We'll take some of those, I guess. So is a normal PCR machine able to amplify COVID-19 RNA?
That's a good question. A normal machine can amplify it, but it cannot do a quantitative PCR, right? So the whole point about a qPCR, what I've showed here, is that you can actually get this sort of a profile, right? This S-shaped profile, which is important to quantify the number of virus copies that exist. You can't do that with a regular thermocycler or regular PCR machine. That's only sort of an endpoint. You can't keep measuring at every point.
Right. Then so of course there are several other questions. I guess we can get started because I also will have some questions when you start talking about these your rapid test, and then we can take some of these other questions. So maybe we can go back to your presentation.
Sure, Rohit. I'll move to the immunoassay part now.
Yes.
So we just went through the molecular, and now we're going to go to the immunoassay, the protein tests, basically. So first of all, it's important to know, I guess, a little bit about the immune response in the body. there's something called the innate immune response, and then there's something called the acquired immune response. The innate immune response is the first thing your body does, right? Like, so even your skin or other protective mucosal barriers, all of this is part of that. You know, it prevents viruses or other, bacteria or other things from entering your body. And even when they do happen to enter, there are sort of non-standard things, non-specific things that happen to where, bugs are chewed up and so on. So that's all part of your innate response. Now, what is known, by the way, is that this COVID-19 is sort of obviously a very clever virus and is able to evade the innate immune response. And that's an important part of its being so infective. It's actually able to get away from your standard immune response, which is why, let's say if somebody, passes a cold in a week or so, they're not able to do that with COVID because of its ability to evade this. And some of these things are honestly being understood as we speak. People don't know exactly why, but it's able to evade what's called the IFN gamma response that's seen during initial infection. But after that, you have the acquired response, where sort of your body is figuring out what this virus is, that it's got this spike protein, it's got this N protein, and your body starts generating antibodies. And antibodies are very specific molecules that are made by B cells in your body that actually bind to these viral antigens, right? So antibody binds to antigen, antigen comes from the virus. So your body will start making antibodies against very specific viral antigens, which could be the N protein, it could be the S protein, it could be E protein, it could be any of these, right? So your body starts making antibodies against all of these things. And like I said earlier, what the body does actually is it first makes, I'll go back to this chart here, it's really important to understand this again, I'll repeat it, that you first start making IgM, and actually also in this case, something called IgA. So these come on first, right? And then effectively your body is optimizing its immune response. And once it kind of understands effectively the virus better, you then get this IgG, which is shown here in red. So the IgM is in green and the IgG is in red. So both of these are part of your acquired immune response. The IgM, as I said earlier, goes away and the IgG stays on typically. So now coming back to this, I think I put a picture of the virus here again, just to mention to you that you've got all of these proteins, right? And the virus, by the way, gets chewed up inside your body. So it releases all of its constituent proteins inside your body. So your body is kind of able to sample the different parts of the virus and generate antibodies against even something inside here, like the N protein. And because there's a lot of this N protein, it's kind of become a favorite, you could say, to actually make, you see a lot of antibody against the N protein. One thing I'd like to mention about antibodies, which is very important here in the context of immunity and such questions. So here on top, I'm showing you a picture of ideally what we'd like an antibody to do. So this neutralizing antibody, as the name indicates, will bind, let's say, to all the spikes on a certain virus and prevent it from binding to the ACE2, correct? So this is what you would hope the antibody would do. And that actually would count as sort of conferring immunity. But what is not known right now is if this is exactly what's happening, right? And we're hearing stories of people getting reinfected and so on. And there are known cases in the past where you have sort of things where antibodies are generated, but not enough of them are generated, or they don't have enough affinity. Affinity is simply how strongly the antibody will bind to, say, the spike protein. So if your antibody exists, but it's not strongly binding enough, that might not be good enough to actually give you immunity. So there are all these second and third order questions that need to be answered. One thing that's interesting from the literature is that the antibodies binding to the spike protein receptor binding domain, the RBD, that is the part of the spike protein that binds to the ACE2 in your cells, were found to be the most effective in SARS. And this is not surprising in some sense, because that's what the virus is using to bind to your cells. So if your body is able to generate an antibody against that receptor binding domain, it is going to be more likely to prevent the COVID from binding to the ACE2. So these kind of questions, again, are being answered in more detail as we speak. So we have already talked about this, so I won't repeat this point again. So here is actually a view of the spike protein. Okay, 1 second. Get this. So this is a view of the of this, of the genome that I talked about earlier, about 30,000 base pairs. And shown here in pink is the spike protein. So the spike protein actually has about 1200 amino acids. So a small detail that I'm skipping here is you're going from nucleotides to proteins here. And every three nucleotides effectively makes one amino acid. So that's why you have a reduction in number here. But ignore that for a second. You have 1200 amino acids making this spike protein. And in fact, as you all know, there's a lot of conspiracy theories going on right now about where this virus emerged from and was it man-made and so on and so forth. And one of the strongest pieces of evidence that it is not man-made is actually coming from the spike protein. And I think the most sort of experienced virologists in the world believe that this is not man-made, because when they study the spike protein very closely, what they have found, and I'm showing that sequence right here, this is a sequence of amino acids. It reads PRRAR, okay? And if you go back to my table, which I showed you earlier, you can kind of use that to find out exactly which amino acids those are. And I can tell you R is arginine, and it's a basic amino acid, right? So it is very known, it is well known that this a polybasic cleavage site, right, something which has multiple basic amino acids together, is actually the virus is using an enzyme in your body called furin to kind of cleave it and then allow it to enter your cells. So this particular sequence is very, very important. And also this receptor binding domain here is very important. So these are two, in this 1200 amino acids of the S protein, these two are very, very critically important to the functioning of the virus. And if you see here, there are these blue things, receptor binding domain residues. So you can see 1, 2, 3, 4, 5, and 6. These 6 actually come together because this is not a linear, it's only linear here, but imagine it folds together like a string. These can actually all come much closer to each other and they form a pocket, okay? And that pocket actually helps it bind to this ACE2. Now, what people have found is that that pocket is very crucial to this virus, and it matches exactly with something seen in pangolins. So that's where this whole bat to pangolin theory has come from. That is a combination of a bat and a pangolin virus, especially this receptor binding domain, which gives it this ability to bind very nicely to ACE2, can't have been created by man. Effectively, this needs to explore parameter space of such a large amount that you cannot imagine human experiments effectively coming up with this, right? It's only natural selection that could come up with this combination of a nice receptor binding pocket that was not predicted previously by, say, computation. So this is sort of the short answer to why people believe this is not a man-made virus. But anyway, we'll come back to our diagnostics here for a second. So I think these receptor binding domains, again, even from an antibody point of view, are very important. You want your antibody to go and bind to these very same molecules so that it gives it that ability to block the virus effectively. So now, if you could actually, how would you do this immunoassay? The principle is very simple. You isolate the spike protein or the N protein of the virus, and that's what's shown here as the capture antigen. So that's actually coated on a surface, on a plate here. So this is an ELISA plate here on the right, and on the bottom surface of this, you coat this N protein or the S protein. Now, as your body is actually recognizing these things, a patient that's actually been exposed to the virus will have antibodies inside them, and these antibodies are typically shown as these Y-shaped molecules. These antibodies will actually then bind very specifically to that spike protein, let's say it is here. And then you have another secondary antibody which has got an enzyme on it, which produces a color. And that's simply so that you can actually detect this binding event. So this antibody recognizes this antibody and then produces a color change. So the more blue you are, the more amount of antibody you have in this plate, right? And that's actually read by some spectrophotometer. And you can then say how much IgG or IgM. So these molecules could be either IgG or IgM, and you could actually quantify this on an ELISA plate reaction like this. So just again, what you need as the reagents for this test are actually the viral proteins, the spike protein or the N protein. And these are made in the lab by effectively taking, again, the cDNA, that is the DNA of the virus, putting it inside an E. coli or an insect or some other species and actually culturing it in the lab and making large amounts of the viral protein without actually needing the virus itself. So this is a very standard technique that's used in molecular biology when you want to make large amounts of protein. So that's what people are doing. And there's a ton of companies out there already making these S and N proteins, which have been, let's say, made in E. coli or something, and they're supplying them to people who want to make tests. The other kind of test that has got a lot of attention is the rapid tests. And the rapid tests obviously have a lot of value, especially in the Indian context, because they can be done very simply at home by a relatively untrained operator with a finger stick of blood. So that's why people are rightly attracted to the idea of rapid tests. They don't need instruments also. The rapid tests also work on a similar principle to the ELISA. You basically have this N protein or the S protein of the virus, which is coated with some dye. The dye is usually a gold particle that shows up as pink. And then when you add your sample there, your sample contains antibody which will bind to this N or S protein because you have antibodies against it. And that complex migrates downstream where it binds to this anti-human IgG, anti-human IgM, and produces a line like this. So very much like the home pregnancy test. So the components of the ELISA and the rapid test are the same. You need the spike protein or the N protein, and you need the anti-human IgG and the anti-human IgM. And that creates a line. And in fact, you could do some nice thing where you say, if it's only IgM positive, maybe it's an early infection. If it's IgM and IgG, you could say that maybe it's sort of a 14, 15-day infection. If it's only IgG, you could say that you've passed the infection and you've retained the immunity. This is sort of typically how people do this. There are some complications again with COVID, but that in theory at least would be how these tests would work. So here's the important point. I think right now, people don't know if exposure to the virus actually means you've acquired immunity. For most other viruses, that's true. But with COVID-19, there is a lot of confusion and doubt about this, and hence the fear that just because you've been exposed and just because you've generated antibodies, it does not mean that you have actually acquired immunity. This is really, if I had to say one thing in this presentation that you should all take away, that's all. That's the most important point. That exposure not necessarily equal to immunity at this point. We don't know. Maybe it does, but we don't. There's a lot of details we don't know at this point. So how do we actually deal with that? How do we improve these antibody tests? So the mere presence of the antibodies does not confer immunity. So what actually helps you understand that, and this is only done in a lab, it can't be done routinely right now, is something called a serum neutralization test, where you collect the antibodies from a patient, and then you grow the virus in a culture, and you see if those antibodies are actually able to neutralize the virus, which, as you can imagine, is sort of really proof of the pudding that your antibodies are actually getting rid of the virus, right? But this is obviously a test that cannot be done very easily in a widespread way. But a lot of researchers are doing studies like this in labs right now to determine exactly which antibodies confer sort of this neutralization, which is the key point in terms of being able to get immunity, all right? The other thing that we don't know is how long the antibodies last. So with these coronaviruses and even with SARS, I think people have seen that unlike, let's say, smallpox, where once we've all been vaccinated, it lasts a lifetime, literally 90 years or so. But with these coronaviruses, it seems like these IgG levels rise and then drop, and then they get to sort of insignificant levels after a few years. And maybe for some people, it might be a few months. We don't know all these questions. So how long does the immunity actually last, even if you have it? And unless we know that, again, the same question comes back, that can you give this immunity passport when you don't know how long the immunity lasts? So these are some of the challenges that need to be solved with the antibody tests and being able to draw conclusions that they actually give you immunity. So what's the summary of all this? I would say in some sense, this is a study in China where they used RT-PCR. And you can actually see the sensitivity even from day 1 to 7 is 67%. It's not that great, honestly, for an RT-PCR. And again, these are tests that have been developed in a hurry and so on. They'll probably be getting better and better as we speak. But the interesting thing to take away from this table is that when they measured the antibody, which is the IgG plus IgM, especially after day 8, they got very good sensitivities by measuring antibody. In fact, as the body probably started clearing the virus, the sensitivity by RT-PCR started dropping, but you had really good sensitivity by antibody. So right now, I think what you can say is an antibody test done well can tell you whether somebody has got the virus, has been exposed to the virus. What it cannot tell you is about the immunity at this point. So the authors recommend, and I think this is becoming sort of, a lot of people who are experts are saying that some combination of PCR and antibody tests is the ideal protocol going forward to determine all the answers that we need as a society. So here's a table to quickly understand, I guess, all of these tests against each other, how they stack up. So, affordability, right? We want this to be really cheap. So molecular tests don't score very well there. The RT-PCRs are kind of expensive. They're about 4,500 rupees each in India. And even the reagents can be quite expensive. On the other hand, rapid tests score high because they're quite cheap and you can make them in the millions quite fast. ELISAs are somewhere in between. Sensitivity, specificity, obviously, like I've mentioned at the beginning of my talk, the molecular tests are the best, especially early on in the infection. They can catch it better than anything else. Rapid tests, not so good. In terms of being user-friendly, again, rapid tests score because they could be done by, let's say, ASHA workers or not so trained technicians out in the field could run these. Molecular tests need fancy labs and, you want to protect from contamination and all these kinds of things. Making it equipment or instrument free. That's again something that the rapid tests score very well on because you could just read it visually. The RT-PCR obviously needs quite a fancy equipment to run. But there are some other considerations like batch size, right? Like in a context like this, you want to be running thousands and tens of thousands of tests. Now things like ELISAs and molecular tests have very nice automated instruments built to run this like factory production, right? Like run literally thousands of tests a day, automated like 24/7. Whereas rapid tests, you've got to have that person who's trained, who's got to go take your sample, etc., etc. So the batching is a little bit more difficult to imagine happening there. Turnaround time, these can be as fast as 15 minutes, so they score well. These tests need at least an hour, if not two, both the molecular and the ELISA. So there's a lot of trade-offs here, which I've tried to summarize in this table. The next thing that I want to talk about is just some important trends going forward. Ideally, you want to combine best of both worlds, and Rohit already talked about LAMP. But if you could do basically LAMP or something else which enables point of care PCR tests, right? So you don't need a fancy lab, but you can do this PCR test and get its sensitivity, but in a much more distributed location, like a slum or a smaller nursing home or whatever, then you would have a lot of advantages. So Abbott now, I think many of you probably read this, they have an excellent platform. And I'd like to make a small point here. It's not like Abbott made this in 3 months. Abbott now has been around for the last 5 years detecting influenza in the US. They made a COVID test on that very fast, right? And which is pretty incredible, right? But the point to note here is unless you've invested the time and years and effort into making a working platform, and this takes honestly a decade to do, trust me, then you don't at a time like this suddenly have this platform available to actually do testing. So that's an important point. Here in India, we have our own company, MolBio, which has a platform called TrueNat. And I believe some states are already procuring this, which is also a point of care molecular test that can be done without access to very fancy lab facilities. So we have an Indian company that has got a PCR test that can be point of care. Coming to my own company, Achira, going forward, one of the things that we feel is going to be very important is, can you simultaneously test for dengue, COVID-19, chikungunya, SARS, MERS, etc., sort of a fever of unknown origin panel, right? And you can just imagine that this is going to be very relevant 6 months or maybe years from now, that people have some sort of fever or some sort of overlapping symptoms. They're scared. They don't know what it is. Can you do a multiplexed panel that is simultaneously detect for everything? And that's exactly the kind of things that we do. So we have a multiplexed immunoassay platform that could do multiple detections at the same time. So maybe can we do all of these together is the kind of thing that we are looking at. We also have other people, like there's a company in Pune I know that has a multiplexed LAMP where they did Zika, dengue, chikungunya together, right? This is again something which can bring LAMP and multiplexing together for sort of point of care applications. So I think a lot of things to look forward to in terms of new developments. The other important point is that post-vaccination, even once the vaccine is developed, whenever that is, we'll have to test whether the vaccine is effective, because likely it will work for some people, not work for everybody, and so on. And the only way you'll know if the vaccine is working is by measuring again antibodies. So these tests will continue to be important even through that phase once you develop the vaccine. So I'd like to summarize the talk at this point. So number one, molecular tests offer excellent sensitivity and are the gold standard for detection, especially during the early phases of COVID-19 infection. So 0 to 7 days, I think nothing better than a molecular test in terms of sensitivity. The antibody tests can be cheaper and they can be easier to deploy, like the rapid tests. The sensitivity is very good if you are talking about 7 to 9 days and after infection. And they can also potentially, again, I stress that again, the potentially tell you about patients who have acquired immunity once we get the reagents right going forward. So ideally, a combination of the two, the molecular, the gene tests, and the antibody tests will be required in the coming years and months to effectively test both for COVID presence and for acquired immunity. The science behind testing for immunity is still evolving and not fully understood. So I think WHO actually came out with a statement last week saying, we don't know, basically. You have to wait to know when we can say that such and such test will actually test for immunity. We don't yet know. Last thing I'd like to mention is this is really about testing for the virus and immunity. But in addition to these, there's a whole bunch of other diagnostic tests that are very crucial, especially in the ICU setting. You've all probably heard of something called the cytokine storm, which happens in patients that are critically ill, where your body is basically fighting the infection, but basically going berserk. And being able to test the elevation of these cytokines in people's blood samples, doing it fast, could help identify critical patients in the ICU setting and people that need care urgently. So there's a bunch of other tests like that that are also very relevant, I would say, in the COVID testing scenario, beyond just testing for virus and immunity. So with that, I will stop and pause here and we can take forward the discussion.
Yeah, thank you very much, Dhananjaya. Very interesting talk. Let me ask you some questions that have been posed in the QA, and then I can ask you some of my own questions. Okay. So, let's start from the bottom. There's a question which says, somebody likes your the test that can do multiple causes, the fever test panel that you said.
Yeah.
Now, is this commercially available now?
No, it's in development, Ashok, because that's something that, we'll have to put a bunch of things together still. But the hope is that to have something like that in months.
Yeah, and I think that's a wonderful way forward because, many companies are now investing in doing these multiple, detection because, it might actually lead to a much wider platform technology than what we currently have.
Absolutely.
So there was, let me see. There is a question that says that we know that viral particles have been detected in other organs like kidneys, etc. So are there tests to detect virus using different tests that targets other organs? Let me frame this a little differently. One is, can you use blood serum, plasma to do a rapid test?
Yeah. So
antigen. So let me rephrase. For an antigen-based testing.
Okay. Understood, Rohit. I think that's a very interesting question and rephrasing. In theory, yes, that you could directly detect the virus, but this goes back to a point I made at the beginning of my talk, which is that if you only have a few copies of virus around, unless you have a tremendously sensitive technique, which is be able to identify just a few copies of the protein, you will not likely pick it up. The DNA, RNA tests are different because of the PCR's ability to amplify the reaction. There are sort of cutting edge protein technologies that are trying to get to these kinds of femtomolar, very low concentrations, but they're not very widely and easily available at this point. So it would be a tricky thing, but I think an exciting thing if it worked.
Yeah, I think it's difficult. So from my perspective, I think virus when it appears in the blood is already too late then. So you should have picked it up much earlier. But going back on a different thread here, Dhananjaya, you talked about antibody-based rapid tests. What are these antigen-based rapid tests?
Well, I guess the antigen-based rapid tests will sort of, I'm assuming, Rohit, will start with the swab samples and try to detect the viral protein directly after lysing the virus. again, if you have enough copies of virus, somebody's in acute infection or whatever, you might even pick it up like that directly, direct antigen tests.
So
But then
that's what,
it all,
this is something that South Korea tried out, and had a lot of success, Dhananjay. And, I had a chat with Dr. Swaroop also regarding this, and she was very excited that, if somebody can make these rapid antigen-based tests, and these are, sensitive and specific to the numbers that we would want them to be, then, this could be the way forward for all these testing kiosks that, South Korea did and, was able to contain the infection really well.
No doubt. I think if we can demonstrate that performance, Rohit, there's no doubt, because now you are directly detecting the virus, right? You're not detecting the host response, the antibodies. Right. So if you can do that, there's no doubt that that would be a very exciting test.
So maybe, your company could have a look at it.
Absolutely. Thank you for the suggestion. Something
okay. So there is another question. Let me see. Do the ELISA test and the 3-strip home test work on the same antibody detection technique? I guess you answered that.
Yes, broadly, yes.
So there's another question, and this is a very relevant question. They say in order to test large group of people for COVID-19, is it possible to test, say, 10 people altogether? So if all of them are negative, you can economize on the test.
Yeah, the
positive,
pooling.
You can do small to finally isolate the positive. Yeah, the pooling.
Yeah. No, I think, by the way,
So
pooling.
I guess we are.
Go ahead, Rohit. You go ahead.
Yeah. Go ahead. You go ahead.
Yeah. So I think pooling is something certainly on the molecular tests. By the way, pooling is already done routinely in India for blood banking, right? Because when we want to test for HIV, hepatitis B and C, those tests are also very expensive. So it's often blood bags, samples are pooled before being tested. And I think 10 at a time is quite common. Now, antibody tests being pooled is something that I think is a little bit more complicated than the molecular test being.
I agree. So one question I had, Dhananjay, you talked about this IgM and IgG, and IgG, is actually showing you a long-term, kind of a response.
Yes.
Right.
Yes.
So is this, so if somebody gets an IgG response in the rapid test, does this show a response to other, coronavirus infection, or particularly the COVID-19?
Yeah, no, that's the very important question, actually. And you need to demonstrate in your testing protocols that it is not doing that in order to say you have a good test. And I think these are exactly the kind of things that are probably not being done right now in a hurry to push out products, correct? That people are not testing for all these, because there are, like I showed you earlier, a lot of sequence homology in the S protein, let's say, between the earlier SARS virus and this COVID-19. So it's possible that there could be, I guess, antibodies, that are cross-reacting in that case, and you don't know. And this is where it will come down to getting your reagents right. I think making those reagents very specific to COVID-19 will be the key here.
Yes. And I guess, for all these antibody-based tests and the antigen-based tests, raising the right kind of antibodies, the monoclonal antibodies, and their purity is a really crucial thing, right?
Yeah. So you're talking about the direct antigen test, right?
Right. Yeah. Even for the antibody-based test, you will need the anti-human IgG or the anti-human IgM, right?
But those, Rohit, are quite routinely available, and those are not considered problematic reagents. So you'll have to get the NMS protein right, of course. Right. But the anti-human IgG, anti-human IgM are not. I mean, they're quite routinely available.
Okay. So that's a good answer then. So somebody is asking, does Strand Life Sciences have a role to play in this, using bioinformatics? I guess, they will, right?
Yeah, I mean, I think anybody who is, and obviously Strand is, one of the leading companies in India in terms of, I guess, doing lab testing now. So I guess if they develop their own test and so on, I'm sure they would be able to add a lot of value. Better ask to them.
So there was another question. I don't understand this fully, but somebody has asked, can we use gene editing techniques targeting the RBD?
Are you referring to CRISPR, Sonal? guess let me just.
Yeah, I guess that's probably.
Okay. Yeah. So I think okay, there are people doing R&D on using CRISPR to do it. So I think the thing with CRISPR, which is not clear to me at least yet right now, is whether it can avoid the amplification. The whole, I mean, the painful part in some sense of PCR is like the amplification, right? So if CRISPR is able to avoid amplification and still detect, then it would be great. But whatever I've seen so far of the CRISPR methods is that they also need the amplification to happen first. And then it's sort of CRISPR is used to do the readout, which I personally think is less exciting. But I think CRISPR has the potential to get rid of the amplification, which would be great.
Right. And you know, sitting in some of these committees, we have come across several companies which have put forward this CRISPR-based, you know, test, and you know, hopefully, you know, one of them will have, you know, a solution soon.
Yep.
There is another question to you, Dhananjay. You know, your company has been working a lot with microfluidics. You know, you have a terrific setup there, and how would you make a rapid test on a microfluidic platform?
Yeah, good question again, Rohit. I think, see, in some sense, the test principle is not going to fundamentally change, right? You need a surface on which you put antigen or antibody. You need a flow to happen. You need a detection reagent, and then you need to pick it up, right?
Right.
So the way I think about it is you should use microfluidics to do something which a lateral flow cannot, because you can't beat a lateral flow at its own game, right? I think mass-produced cheap tests for IgG, IgM, I think doing that on a microfluidics platform, I don't know how much value honestly it'll add. But the kind of thing that I suggested earlier, being able to do much bigger multiplexed panels, maybe being able to do direct antigen testing by being more sensitive, I think those are the areas that I would say if the microfluidics can help, that will add a true value over and above what already exists.
In fact, you know, I was thinking, Dhananjay, why not do something for the LAMP protocol, you know, and, you know,
Absolutely.
combine your microfluidics with that.
Absolutely. And I think that would be a great idea too. If you can, I was only thinking about the immunoassay right now, but absolutely, if you can do a LAMP and sort of make a cartridge which can, you know, hold a chamber of this fluid, heat it up, and 60, hold it for this thing, and yeah, absolutely.
Yeah, that's the plan, you know, for, you know, I think that would be something that your company should, you know, in fact,
Sure.
explore.
Absolutely.
So, you know, there are some other questions. So, you know, there was a question in which somebody asked if, you know, some, you know, antibodies are taken out from a recovered patient, what all parameters are checked before giving it back to another patient, I guess.
Oh, this plasma therapy stuff.
Yeah,
right?
yeah.
I should say that I am not a clinician and I don't want to say something.
Yeah, I know.
This is sort of a slightly controversial and evolving topic. So I would prefer to differ on that question. Actually, I don't know.
I would too. You know, it is something that is. But you know, going back again to the cost of these tests. You know, right now the RT-PCR tests are about, you said 4,000, 4,500.
Correct.
And you know, somebody is asking, is it available? You know, these rapid tests are available. Are they available at affordable rates in India?
Yeah. So I mean, probably the price at which the rapid tests are being sold to the labs would be on the order of 500-600 rupees right now.
Right.
And so to pay to the patient, it might be say 1,000 or 1,500. It should be a lot lower, honestly. There's a lot of I don't know, you know, commercial activity in this space, as you can imagine, because of the demands. Yes. And so there's a lot of market economics which has taken over things. I think ideally we should have a lot cheaper, like 100 rupee type of testing available for mass. But it's a little bit of market economics at work right now also, and demand supply shortages.
And there was another question, you know, from Professor Mishra, you know, how would you rate the quality of testing in India compared to that in Western nations?
Dr. Mishra, why do you want to get me into trouble?
So you don't want that on record, huh?
I'm joking, but look, I think honestly, anybody I think the ICMR has got like great labs. We've got these RT-PCR machines set up, and we've got gold standard kits there. So I think maybe the numbers of tests we all feel should go up, but the tests that are going on itself, I think, especially if they've passed all of the stricter standards in terms of measuring their sensitivity, specificity, I mean, I see no reason to disbelieve them, I guess.
I agree. And, and of course, all of us are waiting for this, COVID-19 to finally go away. And we really don't know when it will, but, how long do you think we are going to see before a vaccine comes up?
Yeah, that is a very controversial question right now. I'm sure all of you are following the news there, that there is a company called Moderna, which has promised to the US government that they will have a vaccine in 2 months or something like that.
That's unbelievable. Yeah.
Slightly unproven technology, which is basically an mRNA-based vaccine, where effectively the spike protein that we talked about earlier will be made inside. You'll make that inside your body. Train your body to make that spike protein, and then the body will respond to that. That's the strategy the US is going after effectively. And this Moderna company was just given a $483 million emergency grant. So it's absolutely crazy. But I think a lot of people in China are also trying, and I think BIRAC is also looking at a number of interesting vaccines.
Yeah, there are several, yes.
So I don't know enough about vaccines, honestly. I'm just only commenting and reading other people's stuff.
Okay, so I think that kind of covers everything that people asked you, and it was a wonderful talk, Dhananjaya. But more importantly, we got a feel of what is happening, and then what are the possibilities. So let me once again congratulate you on what your company has been able to do in the past, and we hope that you'll be able to come out with a much more sensitive, specific, and affordable panel that you just described in the near future.
Sure. Thank you. Thanks very much, Rohit. Appreciate it. And thank you for the wonderful moderation.
Thank you. Thank you very much.
So I just wanted to say thank you, Dhananjaya and Rohit, for your time and for so readily accepting to speak at this webinar. This has been a very engaging discussion, and it's spread over a wide range of issues to do with testing. So thank you again. And big thanks to our participants today, to the audience, and for all your questions. Please stay tuned. We will have our next webinar on Saturday next week, and we look forward to seeing all of you there. Thank you.